Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to the start of MDDI's Focus on Fundamentals, Big Changes in Medical Device Biocompatibility, sponsored by Nelson Labs and broadcast by Informa. It's day one, and we'll be discussing updates coming to ISO 10993-1. I'm Omar Ford, Editor-in-Chief of MDDI, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. Also, if you haven't already, be sure to click the Register Now button in your viewing window and join us for the remaining 2 days of this Focus on Fundamentals series. And no, day 3 will conclude with a panel of all our series speakers addressing your questions. Lastly, if you are experiencing any technical problems, please click the Help widget found at the bottom of your screen, or type your issue into the Q&A area, and we will be glad to offer one-on-one assistance. Now, on to the day 1 presentation, updates coming to ISO 10993-1. Discussing today's topic is Audrey Turley, Director of Lab Operations Biosafety at Nelson Labs. To learn about our speaker, please visit the speaker widget. Audrey, over to you.
Thank you, Omar, and thank you to everyone that is joining. This is a hot topic. There's a lot to talk about, and so I do want to dive right in and go over our agenda about the updates coming to ISO 10993 Part 1. I understand that there has been a lot of chatter and publication and already webinars and discussions about these changes, and I don't know that we can ever talk about them enough. And if you've already joined several of those prior to this, I know I have. I've read lots of posts from my fellow colleagues in the industry. And I think even if you've joined and watched and read some of these other things, it's really good to continue to join, because you never know who's going to give you that one piece of tidbit that you just missed or needed to understand a little bit better. So if you're like me, I join all of these just so I can hear what's going on and making sure that I'm prepared to help any further, just help our medical device manufacturers adjust and tackle this change as it's coming forward. So the agenda here, I've just outlined some of the changes I want to talk about. I'm not going over every single change. I'm trying to pick the ones that I think will hit the hardest. And depending on your role in the industry, as a regulatory person, or maybe you're QA for a device manufacturer, or you're the engineer, these will all hit just a little bit different. And so I'm hoping as we talk about the changes that you will think about how this impacts you in your individual role at your company. So first, I want to talk about the alignment here. So the document is more aligned with ISO 14971. with a firm conversation about hazards, harm, and risk. So I put up some brief definitions of these. I give a few different definitions. I think we can define these multiple ways. But for hazard, it's a source of biological harm that's relevant to the intended use. I know some of you are already saying, well, what about foreseeable use? We'll get to that as well. The harm is injury to humans from an adverse biological effect of the medical device. So this is due to exposure to the hazard. And then the risk is the combination of probability and of occurrence and severity of harm. So if you've ever heard a presentation from me before, some of this conversation is the same, especially when we talk about risk. So from my perspective, we're actually not having a new conversation. We've been talking about risk for medical devices since the 90s, when we updated the name of the 10993 document to include risk. So we're not changing or having a new conversation, but we are having and including more precise terminology, and we are aligning with the industry as far as risk assessments. I pulled this figure from ISO 14971 because it helps me think about hazard, harm, and risk. I think it's really important, especially for me because my role is a risk assessor, that I wrap my head around these terms so I'm very clear in my documentation. So this is from figure C1 in the 14971 standard, where when we talk about hazard to harm, there is some consideration about a hazardous situation. And this again is where we talk about intended use, foreseeable misuse, bioaccumulation. It's really hard to talk about these topics in silos, so I'm going to weave them throughout the presentation today so you can hear how I will be applying these in our risk assessments. So when I look at a hazard again, that's the potential source of harm. Then we have to have a sequence of events in order for a hazardous situation to occur. And that really stems from exposure. So we think about foreseeable misuse, we think about bioaccumulation, we also think about intended use of the product. And that will lead us to determine if harm is possible, and then what is the severity of that harm, so that we can then calculate risk. So I came up with a really very generic example. So please don't criticize. If you want to use it, go ahead. I just thought of this really generic example here, thinking, okay, sharks are a hazard. But we need to think about the situation first. So if I'm in a pool, not so much a hazard. When I was like 5, I still was afraid of sharks in pools, but I'm old enough now to know sharks are not in pools. But if I'm in the ocean where sharks are present, then I have a hazardous situation. So the harm possible in the pool is none. But the harm possible in the ocean is medical care to death, or of course none, but we have to consider what the harm is. So that's just my really generic thought about how we want to think about hazard to harm, and then we will go to risk. And in this case, if you were talking about sharks, like that's a lot easier to calculate and exposure. One thing, I'm just going to go back to the previous slide. One thing that's difficult about this, especially when we think about medical devices, is that can we really predict and quantify the probability of a hazardous situation leading to harm? So if we think about, okay, there's a carcinogen on the device. That component will have patient contact. The carcinogen, may not leach out in the patient, and it might leach out to the patient. So there's these different scenarios for us to predict. So while we may be used to using 14971 and being able to calculate these things very thoughtfully because we have control over the manufacturing process or over the engineering of the device, we don't have that same thing from my perspective. I don't have that, as I'm not a manufacturer, but I'm an assessor. And so there has to be some really thoughtful consideration of how we calculate whether harm can occur or not. And this is where we'll look for additional data. And I'll talk about that throughout the presentation. But this is where the conversation is very similar. Because if we don't have data to show that there is no risk, then there's still a potential for risk because it's unknown. And so we want to gather data so we can mitigate that risk. Okay, so this is examples from the standard talking about hazardous situations. And I find this very beneficial. I'm curious if a lot of you have skipped over this, maybe thinking, oh, I can just go straight through this. I want to know how my testing has changed. This really helps us understand what will change. So hazards that lead to harm. So these are hazardous situations, hazards, and then the situations that lead to harm. So there can be presence of released harmful additives. So again, released is the key word. Presence of hazardous particles. So particles being present is also a hazardous situation. And presence of a potentially toxic compound. Hazards that do not lead to harm. Hazards are contained in a non-degradable material. So maybe you have a colorant that has a carcinogen in one of as part of the makeup, but it's in a matrix where it doesn't leach out, and you can prove that. Another one that doesn't lead to harm is that toxic compounds are hermetically sealed inside the device. So then there's no patient exposure. So I really think that those are important examples for us to consider when we go from hazard to harm to risk. Identifying if harm can occur is going to be critically important. And that's where we will apply a lot of our conversation today when we talk about some of the other changes in the document. Foreseeable, reasonably foreseeable misuse. I think there has been a lot of discussion about this. We've also been asked as a company, hey, can you help us, understand how we would define this? So I want to talk about what I gather from the standard. So the impact is here that you have to consider reasonable foreseeable misuse when you categorize the device. This is not something that you can think about after you've categorized it. It has to be included in the very beginning. So the definition from the standard is it's a use of a product or system in a way not intended by the manufacturer, but which can result from readily predictable human behavior. So when we think about how do we put a cap on reasonably foreseeable misuse, because there's a lot of, we could be really dramatic about the misuse, but again, I like that the definition includes some readily predictable human behavior. Like, we're not going to have the administration desk at a hospital be performing surgery, right? That's not readily predictable human behavior. So we have to think about what could happen in the clinical space with the device and with those that are using the device. So this can be intentional and unintentional. So it could be either. So we think, I've given some examples, like reusing a single-use device, using a device longer than prescribed, incorrect application. Maybe you apply it too loose or too tight. Using adult devices on pediatric populations. These are just generic examples that are provided in the standard as well. And I want to clarify where the responsibility lies here. It is the manufacturer that will and shall document reasonably foreseeable misuse. And so when I think about defining this for devices, I think that, okay, number one, it should be the experts on the device, which is not going to be myself from a testing laboratory. I'm an expert in the science and the assessment and the risk, but not from how the device will be used or how the clinician will apply the device or what training is provided. I know from some of our customers that consulting with a medical doctor or a clinician who's used your product or has helped you develop it is where you would start to have that conversation. And this should be documented within the risk management files according to ISO 14971. So again, part of the design history file. The key takeaway here is that device categorization might be changed for future submissions. So, we're all thinking about how do we apply this as we move forward, and we'll be touching on that. Jenny's really going to hit that on day 3, and Dr. Lauer in her conversation, but it's hard to not think about that now as we go through the changes. So that would be one area to evaluate for your own devices and your own documentation where you would expect that categorization would change. And Jenny will also talk about how we assess that. I think I have some information in here as well. The next thing to consider, again, this is all up front if we're trying to decide, are harmful or hazardous situations that could occur. So the life cycle evaluation that was added. So again, the definition here, I love to work off of definitions because it puts us all on the same page and hopefully keeps us within scope of expectations. So the definition is a series of all phases in the life of a medical device from the initial conception to final decommissioning and disposal. So this is not new for us to think about, especially in biocompatibility in general, because this has also been included in MDR. So we've been thinking about this for other country submissions. What remains unchanged? You still have to consider your materials and make sure that they are fit for use of the device. And you still need to consider the potential for the materials to change during transport, storage, or use. Before, this was summed up in one sentence in 4.7, I believe, from the current published version of 10993 Part 1. Now, this is several paragraphs long, about half a page in the new standard. What is new is that there's a specific component talking about reusable devices, stating that they shall be evaluated for the maximum number of validated processing cycles. There's also a statement in there to encourage manufacturers to review their risk assessments, biological evaluation plans, and biological evaluation reports with post-market surveillance data to evaluate if there's any newly identified risks that had not been captured before. You can also support to address any concern of risk from what you're gathering in your post-market surveillance data. Bioaccumulation. So again, another topic that's going to impact our device categorization and our testing considerations. So some examples of bioaccumulation compounds are PFAS, because we're talking about that all over. They're ubiquitous in the environment. So that's an easy one to point to. And also heavy metals like lead and mercury. So if a substance is known to bioaccumulate, if you have a substance on your product and you know it's known to bioaccumulate, your contact duration time will be adjusted. So if you have a limited contacting device and you have laced it with mercury that's going to go into the patient, you will have to consider a long-term exposure for that based on the accumulation of the mercury. If during the evaluation bioaccumulation is identified, so maybe you didn't know at the beginning, but we've identified something through like chemistry testing, then the contact duration will also have to be adjusted. So this is going to be one of our favorite biocompatibility examples, because we're always going to say, how do you handle bioaccumulation? It depends. It depends on what is used in your device, what material information is provided. So a really thoughtful and thorough material and process evaluation, identifying and being educated about substances that do bioaccumulate will be helpful in your assessment. Okay, this is the big one, I think, other than testing changes, is really being thoughtful and changing our approach to device contact duration. I think this has been different depending on the country of submission, and we are all coming together as an industry here. So what is unchanged are the contact duration categories: limited, prolonged, and long-term. One thing that has changed is that we've taken out brief tissue contact. It used to be called transitory, but now we have a section talking specifically about brief tissue contact. So while you would categorize your device according to one of these three timeframes, you can still talk about the possible risks, so hazardous situation present based on brief tissue contact, and there will be guidances put into this section in particular. So contact duration calculation. So when you have a device with daily contact, you count the days from first to final contact. One thing to remember is that this is not we have to consider if a device is replaced. So if you use one device and use it for a week and then you switch to a new device after 7 days, so day 8 you get a new device, your contact time doesn't start all over again with a new device. We continue device contact consideration. For intermittent contact, so that is with devices that have 24 hours between contact. So add the number of contact days from first to last use. So I just put in here an example, device used every 2 days for a 14-day period. So it's used on like a Monday and then a Wednesday and a Friday and then, a Sunday. So you do your every other day. So there's 48 hours in between contact time. And so we have 7 total days of use. So that changes, again, maybe how we would typically have thought about it, because having that break between device contacts, we would think, we don't have to be as concerned because it's not continuous contact. But that is a change in how we want to think about this. And again, I just put a note here, I'll be a broken record. You must consider bioaccumulation and reasonably foreseeable misuse in your contact calculation. So some really generic examples. Again, I made these up so I would waste time critiquing my interpretation of a use of a device. I just pulled this together for this conversation today. So let's talk about an inhaler for a steroid treatment that's used maybe during an illness. So it's used 30 seconds a day, 4 times a day, 7 days. So really, it's about 14 minutes of use over a period of a week. We would consider that pretty limited contact. But under the new calculation, it's used for 7 days. That's how long the use is every single day. So that now goes into a prolonged contact category. Another example is like an intermittent catheter that's used 3 different days for 6 hours each time. So we're saying 18 hours total. So that would be a limited contact. Now we say use 3 times for 6 hours over a period of 5 days. But again, it's the number of days it had contact with the patient, which is 3. So now it moves into a prolonged contact category. And Jenny will be doing more of these again on day 3. I don't think we can go through this enough. And questions on this is really helpful so we can discuss as an industry how to apply this when it's more complicated than made up devices that I came up with. Okay, I think one of the biggest changes, of course, is that we removed, or that the industry removed Table A.1. So there's no more reference to this as a single table. Instead, we have 4 different tables. However, I really like one table. So for the ease of our discussion today, I still combined everything into one table. It doesn't have all the special considerations and notes. So this is really just for a quick overview, and we'll go into the details. But this really shows what has changed. And when we think about it, although creating 4 separate tables seems like a large change, again, we don't see a lot of change here in the approach and what we need to consider. So just a brief overview here. We've changed how we address systemic toxicity. Instead of calling out acute, chronic, and subchronic, it just says systemic toxicity. Local effects after tissue contact. So it used to be implantation, and now we've made it a much longer name. And then we've changed some of our categories. So we've changed from tissue, bone, dentin to where we say internal tissue. It says tissue, but I'm calling it internal so that we're clear what type of tissue contact. And then, of course, some additional considerations for genotoxicity, which has been a large topic of conversation. So we're going to go through these just slowly. So again, systemic toxicity added. The duration of body contact is an important determinant of systemic toxicity, and more extensive evaluation is required as duration of body contact increases. So instead of us calling out these categories, the ask is that in the documentation, you justify the length of your toxicity that you are assessing for. So if you're doing the animal studies, this will need to be clarified right up front with the laboratory so that we understand the term and length of testing. if this is being assessed through chemistry, you want to make sure you've clarified the worst case use, including bioaccumulation, foreseeable misuse, in the contact time, so that the toxicologist can evaluate worst case exposure. And then implantation tests, so the local effects after tissue contact. So that is a new name, but again, still the very same, similar conversation. Implantation tests can also cover systemic toxicity and hemocompatibility as well. So just a thought there for manufacturers that we want to make sure we're using the tests as much as we can. So when feasibility testing is happening, or large animal testing is happening, or device function testing is happening, if we can loop in some implantation endpoints or hemocompatibility endpoints into those situations where it's really mimicking clinical use, that is most preferred. And so there's benefit in thinking about this ahead of time, before, maybe even before you have your final finished device, as you think about when should we do our large animal testing and how much can we, you want more bang for your buck, to say. Okay, and so we'll go through the talks or when we will increase assessments for genotoxicity. So the first place we see it added is when we have a prolonged exposure to mucosal membrane. And then if we have a long-term exposure, we're also going to consider carcinogenicity for mucosal membrane. One thing to keep in mind as we think about the exposure to genotoxicities or genotoxic agents is that the thought is, if we can leave something behind that maybe with a brief contact wouldn't have a genotoxic potential, but if it stays with the patient, it can create immunogenic environment. And so that's what we've considered when we looked at, because I worked with the working group 1 for 10993 part 1 and the working group over 10993 part 3 worked together to discuss, hey, we need to be thoughtful about when could we actually see this occur. And so it's really about leaving behind, or leaving a residual, or having something pulled out of the device that would stay in the patient. Okay, again, here's our next table, which would be the breached, compromised, or tissue. But I, again, am saying, we're calling it internal tissue. So internal tissues are other than circulating blood. And so this is, we combine breached and compromised with bone tissue and dentin. And again, you see here the addition of genotoxicity to be considered when we have prolonged use. Okay, the next table change is for circulating blood. So this is now one category for part 1, because 10993 part 4 differentiates which type of hemocompatibility testing you need to have. And so if you have circulating blood, we're going to get more of that evaluation from 10993 part 4. So we don't have to define that as much here as we did before. And again, genotoxicity is added when prolonged systemic exposure occurs. So we've added it for B, which is prolonged exposure. And then for A, this is for short-term exposure. However, this applies for extracorporeal circuits. So this is where blood is being pulled from the patient and then manipulated and then pushed back to the patient, so that even if the exposure was short-term inside the device, if the blood has pulled something from that and put that then back into the patient, where it will circulate through the patient's body and could cause a genotoxic effect before it is removed from the patient. Okay, another thing to consider with material-mediated pyrogenicity. So there's been some really good presentations and discussions of why we are changing our approach to this as an industry. Some good presentations and work with Ron Brown and Lindsay and Kelly Coleman. Really appreciate the conversation. I think it was really important to have this. One clarifying point is that this endpoint did not go away. It's just not lumped in, and we're not expecting it to be addressed for every single device. Instead, again, we want a consideration from a risk-based perspective. So you still have to consider if you have a known material pyrogen. So does any material have a history of causing a pyrogenic response? Or it contains a material with an unknown pyrogenic potential. So if we have a new material that we don't know about, or if we aren't sure how materials or compounds may react, then we would want to do at least an introduction or validation MMP test to understand if there is a risk. So again, there's justification possible. So if the final device doesn't present any significant risk for a pyrogenic response, or if you have, again, manufacturing controls to prevent pyrogenic contamination. And that's not just thinking about bacterial endotoxins, because that's evaluated elsewhere, but this is thinking if you have pyrogenic material in your manufacturing line, that you can prevent it from contaminating your device. Biological equivalence. I think this is a bit of a hot topic. It's a good addition to consider. The old standard considered chemical equivalence, which isn't super realistic with our technology today. But I think this is a good use for line changes over time, or similar to like family grouping. This would be really hard to do with a competitor device. So this is really appropriate if you own the device manufacturing. You can speak to the line of manufacturing, all of the process, all of the additives. Then I think there's more of a ground here to have this conversation. I still think this is really tricky. And I would expect this to be really similar for maybe catheters that have different lengths. This is where that conversation is much easier. Again, not a new discussion that we've been having. We talk, instead of saying biological equivalence, we would have said the risks to the patient are identical and we've already assessed them, by this master product or something else. So again, very similar conversation, just with some new or more specific terminology. So I want to think about just some documentation thoughts. So as we've gone into this, how do we capture all this information in a really thoughtful flow? And when we look at the new standard, even when you look at 14971, which lays out the thought process, there's this part of, the BEP, kind of the biological evaluation plan, leaves a bit, it's like an open-ended story. Like you get to the introduction in a story and you get introduced to all the characters, and you know who the evil person is and who the good people are. And you kind of have laid out, okay, now we're going to get into some of the plot here. That's still what will happen here, is that we'll talk about our device information. This is where you'll talk about your materials, the intended use, foreseeable misuse. Bioaccumulation will be in here as well, and you'll come up with your device categorization. And then we'll talk about hazardous situations, and harm, where they will be identified and categorized, and you can really isolate where you expect to have some issues from your materials. And then we'll see, do we know enough? Do we know that, what's the chance of harm occurring from this irritant? Well, we've done a cleaning validation. We have data to say it's not present. So we kind of do this gap analysis. And I use this term for a lot of different offerings or different thoughts when I'm doing risk assessments. But in this case, it's really knowledge gap. Where do we have some unknown risks, or where do we not have enough data? And it's really that's going to decide your testing approach or your research approach, what you need to look into further. And then if we look at everything at the end, so again, this is the introduction. What we have at the end is like how it all wraps up, after all the chaos has occurred, which would be the testing. So I wouldn't say that's totally chaotic, but in a story, your chaos happens in the plot. When you assess your risks in a BER, you're also looking at the same thing, device information. You look at your residual risk assessment, which is now more informed from the data that you've gathered. So you can understand if you actually have hazardous situations occurring, which will lead to harm, which create the risk. And so that's really where we close out this discussion. Again, very similar conversation to what we've had, just in a different frame. And then we talk about risk refinement or risk control if that is needed. And that may happen in a biological evaluation report, but it also could happen in your risk documentation that you use for your device history file. So this is I like to break everything down into three to five, right? That's where we learn, that's what we can hold on to. So I've used this image for years, and I don't think it has changed, but what we include in each step is a little bit different. So for the biological evaluation plan, now we're talking about risk estimation and a gap analysis. What we've done before, but now we have really streamlined terminology for it. For step 2, it's still gathering data. So doing your testing, doing your risk assessments. And for step 3, this was just a summary report of what happened, but that's still what we're looking at. So we say what happened in the plot, what bad character are we not taking care of, or did we get all the bad guys and now we know we're okay. So then we have our summary at the end. And so if you didn't get rid of all the bad guys, there's some risk control that you have to do. If you got rid of everybody, your residual risk assessment or estimate becomes pretty low. So again, same conversation in a more formal standard format. And then I just want to encourage everybody to stay tuned for day 2 and day 3, because we're just going to keep talking about the changes that are coming forward. Nick Cristiano will talk about Class 6 impacting medical devices, and Dr. Jenny Lauer will talk about how to address these updates for your existing products and in-process submissions. So I think we have time now for questions.
Now, before we begin with today's Q&A, please direct your attention to our webinar survey, available on the right of the presentation window. If you close the survey, you can reopen the widget by clicking the icon along the bottom of your screen. Thank you in advance for filling out the feedback form. Your participation in this survey allows us to better serve you. And now, on to the question and answer portion of our event. For this segment, we welcome Jenny Lauer, a biocompatibility expert at Nelson Labs, to join Audrey in answering your questions today. To participate in Q&A, just type your question into the text box located to the right of the presentation window, or click the Q&A icon at the bottom of your screen. If we're not able to answer all submitted questions during today's webinar, we'll be sure to share them with our speakers, who can reply to you offline. And our first question is, for foreseeable misuse, what were the underlying reasons for these changes? Was a manufacturer ignoring misuse cases with their device?
I can take this one. So this is a good question as we think about patient safety. That's the end goal, is to make sure that we are covering our patients for how the device is used. It's no surprise to manufacturers that the devices get used differently than intended. Oftentimes, device manufacturers are aware of that. What we want to make sure is that there's some evaluation of that upfront. So why it came about, I'm not particularly sure, because I wasn't part of that initial conversation. What I will say, though, is that it does make sense to include it when we think about what do we know about the use of the device. So even unintended, we're assessing this anyways in your whole design process, right? Your manufacturing process, what could go wrong? We want to make sure we're considering that for the patient safety overall. So this was actually just a really logical addition, that we should be thinking about this up front and not after it's gone to market and then it's used on patients. And then we already know the doctors, if we already know the doctors will want to use it differently, let's assess for that as well. So we're protecting that patient population.
For bioaccumulation, is the contact duration increased in general, or just for the specific chemical? Any takers?
Jenny, do you want that one or do you want me to take it?
Why don't you take it? You can lead these and, you know, I'll back up, and then Thursday I can lead and you back
okay.
up.
Okay, I like that approach. Then we'll have a thoughtful handoff. Okay. For bioaccumulation, so the question is it for the whole device or just the specific chemical?
Yes.
So if you can separate the chemical from the device, then you can assess it for just the chemical. But if the chemical is an intrinsic part of the device that will bioaccumulate and will have exposure to the patient, then it does impact your entire patient contact duration.
And this next question deals with, will there still be limited or brief patient contacting devices components if it is a stated reasonably foreseeable misuse? Is a device being used longer than indicated? Sorry, that was a mouthful.
Yeah, that's a long question. So I think there are times when you would say it's not reasonable to say this would be left in the patient for, you know, longer than a day, especially if you're having that brief contact. So again, there's room to justify and have a conversation about it. So I think it's important that you're having that conversation up front and not just waiting to see if the regulatory agencies will call that out to you, that you're addressing, hey, we already know this is the typical clinical use. Here's what could go wrong. Here's the chances of that going wrong. And here's where here's the actual risk to the patient. So you can still talk about that being a very brief contact, especially if it's a part that's disposed or removed. Again, you talk about what's the chance of it being left on? Can the device function with it left on? So again, it depends for every device, but you can hear that there's just in me answering the question that there's a lot of places that the conversation can go, and you can direct that based on your knowledge of the use of the device, the intended use, and with consulting with your clinicians about how it's used in practice.
And I would like to just add to that briefly. From my point of view, as the person sitting at the computer writing these risk assessments, I feel like I'll think about foreseeable misuse separately from reasonably foreseeable misuse, because like the 14791 document, people have to list all the possibilities, but that doesn't mean they're all reasonable. So I feel like, then we can take that subset and say, okay, which ones of these are reasonable? And those are the ones we have to really pull into the BEPs.
And along similar lines, for absorbable devices, if you have a device that is absorbed from the implant site in 14 days prolonged, if it takes greater than 30 days to be completely excreted based on ADME data, should it be classified as a long-term device?
This is a great example of where we would consider some changes to contact duration. So when we think about the purpose of contact duration and assessing patient safety, it's for the entire time that they are in contact with the device. So even if it's absorbed, the patient's still exposed to it. So if we have to wait more than 30 days for the ADME process for it to be fully excreted, then we would want to talk about, okay, what's the long-term exposure? So maybe it breaks down, maybe it hydrolyzes into some different components as it's in the body. So then we would want to look at a long-term exposure, like a subchronic exposure for those compounds with the patient. So again, it does change your device categorization, but how you assess that can be different. And it's not saying, okay, you have to say that you are now permanent, so you have to assess all these categories. You have to talk about how those categories will apply to your device and then how you'll address that for the biological effects. Jenny, do you have more to add? Because that's kind of a fun question and a new thing for us to think about.
Yeah, no, I think this is the part of the document that honestly, from my point of view, I find a little terrifying. So I feel like you could work that through in your testing plan without necessarily, like Audrey said, like fully recategorizing the device, bumping it up, but to say, okay, maybe, maybe we could look at systemic, a little bit longer term systemic toxicity to take that into account. So, from my point of view, I think like maybe not recategorizing the device, but testing it with a little bit more thoughtful approach, taking that into account.
So yeah, oh, go ahead, Jenny.
No, I was just saying, great question.
Yeah, so this, you can even hear as Jenny and I talk, we feel differently about it because this is brand new to us as well. I am more willing to change your device categorization because that seems to be the expectation, where Jenny is saying, I don't think you fit in that category. So you can hear that there's some different approaches here, and it's not, in the end, we're still assessing the same risk to the patient. We're talking about the semantics of how we get that done. And it will be interesting to see how this plays out in the conversations. And I think it's important when we come to these meetings, I would love to be able to tell you exactly how this is going to play out. I can give you my best educated interpretation of the standard and how I think it makes sense to approach it. One thing that Jenny and I talk about a lot is that we will keep a focus on patient safety. So we will navigate the semantics, always keeping patient safety first and foremost. And so I think that's important that we think about that as an industry. These changes are to make sure we align in that way and that we can have a similarly structured conversation. It also helps everyone get on the same page a little bit faster. But again, the focus is really for patient safety. So that question about the absorbable is great because we think about the actual contact with the components to the patient is longer than the degradation time. You're right? So we have to wait for that full ADME to occur.
And our next question is, how do you know if a compound will bioaccumulate?
Yeah, so this really is based on what we have as publicly available information. So first you have to identify the compounds if you don't know them. So that's, again, based on your contact time. So we would do the chemistry testing for a longer exposure, and we would look through those compounds, and the toxicologist will identify if it bioaccumulates. If it's a new, just a new material, I mean, we love ingenuity in the medical device. Like, as I'm a patient, I want the newest, best thing, but from a risk assessment standpoint and a manufacturer standpoint, it just means a lot of work at the forefront to make sure your product is safe. Jenny, do you have any thoughts on, I know that they'll do studies for bioaccumulation on new materials, but do you have any thoughts on that?
Yeah, I feel like the crux of the problem with this will be for, when you don't have chemistry data, so we have to think about bioaccumulation for all devices, regardless of the contact duration. So you might have a limited contacting device, but we have to somehow think about this, and we don't have chemistry data for it. So that's the part where I feel like we need to do deep dives into thinking about all the materials and all the contacting materials. And Audrey brought this up earlier that, some of these things we've been forced into in doing the MDR transition. So we've been forced into thinking about these materials, in more depth. So this is kind of the extension of that, and say like, okay, not only do we have to consider, we look through all these chemicals for CMRs, carcinogens, mutagens, reproductive toxins, and that sort of thing, but we have to look for things that might bioaccumulate as well. So I feel like this is one where we maybe just have to do more in-depth research when we're doing the documentation, and then think about like, okay, do we have to consider any of these? And the good thing about contact materials that are used during processing, you could maybe get rid of them. If you spot a problem, you could maybe get rid of it, which is something you can't necessarily do if you're talking about chemistry data and you've got a bioaccumulating thing, you may not know where it comes from.
Will the FDA recognize and adopt the new version of the ISO rule?
Well, if I had a crystal ball, I would tell you it's no secret that the U.S. voted no on this. Not the U.S. FDA, but the U.S. as a whole voted no on this standard for several reasons. And I believe Thor Rollins went over that in his webinar that he did previously. So I won't recount those here. But what we would expect from the FDA and hope is that we have some understanding of what parts they do recognize, which we can go to their website and ask them, you can see what they've recognized. Sometimes that takes time, of course, to get that feedback from the FDA. So in the meantime, it will be our best guess to, again, meet the intent of the standard, because as a global unit, we are moving forward with this. So there will be some components maybe the FDA doesn't expect, but right now, as a company, we are focused on adopting this new method, and we will adjust to the FDA. I think if anything, we may be including more than what they expect, especially if they're continuing to recognize the previous revision of the standard. But yeah, I have no idea on the FDA's acceptance of this right now. There's a lot of conversation going on, and we will want to have some more discussion on the US side for this as well.
And here is an interesting scenario for you all. This question is, my device is used for 30 minutes once a month for 10 times, and then is likely not used again. Is it not reusable even with misuse? And how is that categorized?
So I'm not sure I have, so this is where you get to hear, like if you work with Jenny and I and you bring your device to us, we ask you 1,000,002 questions because we need to understand some of the specifics. And this would be one of those times. If it's used 30 minutes once a month for 10 times, so 30 minutes and then 10 times, I'm not sure how that pans out. Is it 30 minutes, 10 times in a day? So then you're having 5 hours of exposure. So I don't know that I can answer this specific question, but if I'm going to interpret this a little bit and saying, okay, it's used 30 minutes once a month, for 10 months, maybe that's what it means. So the device is technically used for that long period of time. Is it reusable? Is it now reusable even with misuse is what I think the question is. So when we talk about a reusable device, it's really a device that goes through processing after the use. So if they use it for 30 minutes once each month for a period of 10 months and they clean it after they've used it, and then maybe they clean it again before they use it, then that is considered a reusable device because it is reprocessed and it goes through processing. Now, I wouldn't think about it as big as like, these major reused hospital devices. But why I say it this way is because we want to consider that cleaning in the evaluation of the device, because that's part of the manufacturing process. So we will want to understand, are there residuals from those cleaners on the device that could possibly cause harm to the patient, like an irritation or sensitization response? And again, it's not that it would change. If you consider a device reusable, it doesn't change your contact type, but it changes how you prep your product for testing, is the impact there.
So
I apologize if I didn't clarify that, but you can certainly send me an email and I can really answer your question if I guessed incorrectly.
Yeah, Audrey, I found the question. What they were saying is it is definitely not reusable. So I think
it is not reusable. Okay.
Yeah.
So again, this is 30 minutes once a month, 10 times. So it's 30 minutes, but you have a month in between that time. I'm still going to say it's a limited contact device. What would you say, Jenny?
I would say prolonged, but there's the possibility of sticking with limit. So currently that would be limited. Technically that would be prolonged because it would be 10 days of use. But with a month in between uses, there is the possibility that any residuals that you would be exposed to could be eliminated from the body between uses. So this is one, again, this is where like, you know, there a little bit of the, it depends what you know about the device, what is the contact type. You know, I feel like for a device like this, if it's intact skin contact, that would be a different conversation than if it's, you know, got tissue contacts. So yeah, again, it depends, you know, but I think your default, you know, your starting point is prolonged, and then you think about, you know, the ability of things to accumulate, or would they be removed from the body in between the uses? And that's where, you know, that could, you know, make your testing plan change a little bit. That's how I see it.
Yeah, so I think that's great because it's a discussion about a hazardous situation occurring, like where something would remain, so the contact would be long term, you know, longer. So yeah, there's a lot of room to have a discussion on that one.
Yes.
Great question. Thanks, Jenny, for clarifying that.
And I think we have time for just about one more question. And that question is, does that mean limited exposure now is only applicable for devices that are used once in a lifetime for shorter than 24 hours? Does that leave any device in that category?
Yeah, so I think that's a good question, just thinking about how Jenny and I talked about this last one, because I think it means you have to consider and really justify keeping a device in the limited category. I really think that 30 minutes used once a month, I think that's the solid grounds for a conversation about limited category, but it depends, like Jenny said, on how it contacts the patient. If it's skin, the duration doesn't matter. We know that, right? You know, what you assess for is the same. I would even argue if it's, you know, to check your ear or something like that, we could consider that low risk as well. So it just means you need to feed that conversation a little more thoughtfully. I do think some devices will still stay in that limited categorization. I think what will be harder is to keep the brief contact. I think it's possible, but it's just going to be more of a conversation.
Well, that's all the time we have for questions today. Thank you, and we appreciate your time and expertise on today's topic. Thank you to our sponsor, Nelson Labs, as well as to everyone in the audience. We appreciate your attention and participation. Within the next few hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may have not been available to listen to the event. On behalf of our guests, Audrey Turley and Jenny Lauer, I am Omar Ford. We look forward to seeing you again tomorrow.